Last time we talked a lot about reactions, and what we're going to talk about this time is a little bit about basics of biological chemical reactions that are important to molecular communication. So here we talk about, excuse me, actually, let me rephrase this. Basics of biochemistry. So the object of this part of the lecture will be twofold. One is to understand basic concepts of how biochemistry works, and the other objective will be to introduce concepts that we will come back to later when we talk about the biological basis of molecular communication. So this part of the lecture is intended for people who for whom it has been a long time since their last biology class, or for people who have never had a biology class, a formal biology class. We're just going to go over some very basic terminology and concepts so that we have a basis for going forward. So the first thing we're going to talk about is proteins. Proteins are more or less the universal molecule in biochemistry. While not everything is a protein, practically everything functional is a protein. Proteins are basically, or proteins act as nanomachines and carry out the functions of the cell. So within a cell, within an individual cell, there are millions of specialized proteins that are continuously performing the actions that are required to keep the cell alive, basically doing all of the processing that's required to maintain the cell. So it's fair to ask, what is a protein? And to talk about that, first we're going to talk about two types of organic chemical that make up, or excuse me, make up is probably the wrong word, that form the building blocks of proteins. And those are amines and carboxylic acids. If you've ever taken an organic chemistry course, you might already be familiar with these, but the way to think about them is as follows. So amines are organic chemistry blocks that are centered on a nitrogen atom. For the most part, nitrogen atoms make 3 bonds, and an amine is, I'm going to call it a radical group, R, connected to a nitrogen, and the nitrogen is connected to 2 hydrogens. So hydrogens make 1 bond, nitrogen makes 3. So these 2 are sufficient for the hydrogens, and the nitrogen has to bond to one other thing, and it's going to bond to this group. So it's not, what's going on over here is abstract. This can be whatever, but it leaves 1 bond dangling to attach to the nitrogen. And a carboxylic acid also has a radical, not necessarily the same one, so I'll call it R prime. It connects to a carbon now, and the carboxylic acid has the following structure. So oxygen takes 2 bonds or it takes 2 bonds. So it forms a double bond with this carbon. So this oxygen forms a double bond with this carbon. That makes this oxygen complete. The carbon takes 4 bonds. So it has 1 over here, 2 over here. It has 1 additional one to an oxygen. This oxygen needs 1 more bond, and that is to a hydrogen. So what's important about these is that amines and carboxylic acids can form what are called peptide bonds. And what does that look like? So basically, if you have some carboxylic acid, like so, and an amine over here, what happens is when these react, oops, here we go. What happens when these react is that these parts come together, and what do we have here? We have H2O. So those parts form a water molecule. They come off of the respective groups, and what's left joins up. So we will end up with the following. Like so. And this structure is called the peptide bond. So it's well known that this can happen. Now the question is, how does this relate to proteins? Well, in a protein, what we do we start with amino acids. And as the name suggests, an amino acid has an amine at one end and a carboxylic acid at the other. So in other words, it would look something like this. We would have an amine over here, some organic chemistry group in the middle, and at the other end, an acid. Now, if you think about that, if I have another amino acid coming in over here, in other words, I could draw something like this, connected to some different, potentially different group there. And I could have another amino acid over here. Another group here. What do we get? These can all come together. So those will react and form a peptide bond. Those will react so that the water will drop out. The water will drop out. These will react to form a peptide bond. And what you can get, the result, is a polymer that is also an amino acid. So it can keep building and adding amino acids. So two things to notice about this. For one thing, it is also an amino acid. Look here, there's an end here that's an amine. There's an end here that's an acid. This is all connected together, so that's all absorbed into the grouping, into the organic chemical grouping in the middle. And the other thing to notice about this is that these internal parts can all be different. So we can chain together these different functional groups in the middle in basically any combination that we want. The result here, the resulting molecule is a protein. So that's basically how you form a protein. A protein is a polymer that is formed by groups of amino acids. And basically what you can do is you can chain together any sequence of these amino acids in a custom way. So this implies the potential, excuse me, let me back up and just formally define what I mean. So a protein is a polymer composed of amino acids. The complexity and possibilities for forming such a polymer are enormous, especially if the specific amino acids can be controlled at the molecular level. So in other words, if you could put together a custom sequence of amino acids to get exactly the protein that you wanted in exactly the right order that you wanted, you could potentially, you're basically custom building a nanomachine. And that is the basis of genetic biochemistry, is that in fact, DNA and RNA, along with processes called transcription and translation, do exactly this. And this is the basis of biochemistry. It's the basis of life. It's why living things can do such amazing things with chemistry. And it is what we hope to exploit in molecular communication. So let's talk about that a little bit, how that works. So the key observation is that DNA works a lot like a computer. It might seem weird, but really what DNA is a sequence of instructions, as we're about to see. It's a sequence of instructions for building proteins. And then the cell goes off and builds those proteins, and then they build more proteins that build other proteins, and so on and so on. So perhaps it is not exactly like a computer, but the analogy is remarkably close. So let's start off by defining what we mean by DNA. So this is the famous double helix molecule that stores genetic information. And it's really useful to think of it as a recording tape, because that's really what it is. It's storing your genetic sequence, and that genetic sequence is what actually creates the proteins that carry out all of the functions that keep you alive. It's a 4-ary storage medium. And again, you probably know this, but there are the 4 symbols are known. They're known by various names. We're going to call them nucleobases. There are 4 in DNA. They are called A for adenine, C for cytosine. These are just distinct molecules with particular names. G for guanine, and T for thymine. So there's nothing really magic about these. There will be something a little magic about these later, but for now, you can just think of them as a 4-ary storage medium on a tape. And so we mentioned there's a double helix. So bases are attached to both the helixes, but it's not an independent stream on the second helix. It's actually a redundant stream. So there's a complementary strand. On the second helix. This is actually for a lot of reasons. One is error correction. So it turns out that this can do, if you have the redundant string on the second strand, or at least you can detect errors even if you can't correct them. You can see that something went wrong. The complementary nature is that A is complementary to C, and G is complementary to T. So for example, if you have on one strand of the DNA, let's say I have A, C, T, I'm just coming up with this off the top of my head. It's almost certainly totally meaningless. I'm just picking random bases. And so on. So the other strand must be complementary in exactly the same order, except with complementary bases. So A is complementary to C, C is complementary to A, and T is complementary to G, and so on. So this strand is complementary. Meaning, once you fix this side, or once you choose this side is completely fixed and deterministic based on this one. There is something called the central dogma of biochemistry. And basically it works like this. Well, this is actually a very simplified view. There are more complicated views of this, but fundamentally how DNA works is like this. We start with DNA, which is double-stranded. Then that DNA, what I do is I read off one strand of it like this, and I create a single strand. So the single-stranded equivalent of DNA is called RNA. So more or less, you can think of it as an enzyme comes along, attaches to here, reads off all the bases, and makes a copy makes it actually it makes a complementary copy. So it would read off all these bases, and it would make something that looks like this complementary strand. This process is called transcription. RNA is basically single-stranded. It's complementary. It's complementary to whatever DNA was read through transcription. There's one other thing to know about RNA, which is not super important for our purposes, but T does not appear in RNA. Instead, it's replaced by a fifth base, which is called U, which stands for uracil, which is basically similar to T, except it's slightly different. I'm not sure why RNA uses a different base instead of T, but it does. All right, so once we have the RNA, and in fact, this is called famously, mRNA for messenger RNA. Of course, this is in the news these days because many vaccines for COVID use mRNA. So basically, this is what they're using. They're creating a strand of RNA that is then used by the cell through a process called translation to produce proteins. The technique for going from here to here is actually very interesting, and it's basically why I say this is like a computer, because you have this tape, and more or less you read off the tape, and the tape describes the amino acids that go into the protein. So the process of translation works like this. A ribosome binds to the mRNA and starts reading it. Every triple on the mRNA is called a codon. Each codon corresponds to an amino acid. The ribosome for each codon the ribosome obtains that amino acid and binds it to the protein it is synthesizing through the peptide bond. So the way to think about it is like this. We have our single strand of mRNA here. Oops. Remember, it's U instead of T. And so on. This is enough to make my point. So basically the ribosome comes along and connects here. The sequence CAU corresponds to a particular amino acid. And in fact, CAU corresponds, the standard codon table to an amino acid called histidine. That's not super important, but I just want to indicate to you that there are actually standard tables of these. You look them up. They're called codon tables. All right, so the ribosome prepares an amino acid of histidine, then it moves on to the next three. And here we have AGG, and AGG corresponds to an amino acid called arginine. Then it moves on. So basically it plugs the histidine into the arginine, moves on to the next three, AUC. Is an amino acid called isoleucine. And so on. So basically, this mRNA is a program for producing a custom protein that is specified at the molecular level. So what we would have is through the peptide bond, we'd have histidine, arginine, and then isoleucine. And so on. And then we would continue to add custom amino acids. So that's amazing. So what this structure does is from DNA, which records the program, if you like, we produce mRNA, which is a copy of the program. And then the ribosome through translation comes along and runs the program. It basically looks at all these triples and uses them to encode a sequence of proteins, or excuse me, of amino acids that become a custom protein. Specified at the molecular level, exactly like we said at the beginning. And that's the basis, that's the biochemical basis of life. I just want to leave you with one idea here. Well, actually, I'm going to give you a couple of details about this process, which are I find kind of neat. We're actually not going to come back to them too much, but so there are 3 bases per amino acid. And it's a 4-array code, which means the number of possibilities is 4 to the 3rd power. And that is 64. There are actually 3 special bases, excuse me, special codons. That means stop. So in other words, those indicate like the end of the tape. If you hit one of those special codons, then you terminate the protein and stop. So there are 64 possible codons. 3 special codons that mean stop, leaving 61 to encode amino acids. In fact, there are only 20 functional amino acids. Many codons, or excuse me, many amino acids are encoded by multiple codons, but not all. So some amino acids are actually encoded by only 1. Others are encoded by as much as 6. It's actually not super clear why, but it seems clear that there's some sort of error correction step happening here. It would be cool if someone could figure out from the perspective of either communication or information theory exactly why that is, and why some are encoded with a large number and some are encoded with a small number. So that would be a neat question for maybe someone in this class to answer. My last point here is that, okay, you might think, gee, we basically now have the source code for life. We can take DNA and figure out exactly what protein has been produced, and so therefore why isn't biology completely solved? So if we know about DNA, translation or transcription and translation, and codons, it seems like we know exactly the source code for life. So why isn't biology solved? So there's many reasons, many reasons. It's far more complicated than I just explained to you in the last half hour, which should be obvious, frankly. People dedicate their entire lives to this. But one of the biggest problems is what's called the protein folding problem. Which is given a sequence of amino acids what shape is the resulting protein. This question historically has been extremely hard. In other words, it's basically like reverse compiling a program and trying to figure out what exactly it's doing just by looking at the machine code. But recently, you've probably heard in the news, there is this advance by Google using machine learning techniques, and it's called AlphaFold. And this has promised a breakthrough. They can predict the shape of the protein with incredibly high accuracy, far more than anyone thought was even possible, far better than any contemporary technique. So in fact, this part of the problem, which many people have thought was the hard part of the problem, the protein folding problem, looks like it could be on the cusp of being solved. So this is actually quite an exciting time to be interested in communications and information theory at the intersection of biology, just like we're talking about here. So again, this has been a very brief half-hour introduction to the very basics of how biochemistry works. So this gives you enough of a background that we can continue talking about biological concepts for the context of molecular communication. But there's I want to emphasize there's plenty more to understand about this, and there's plenty of opportunity for communications and information theory to do interesting research in this space.